The protein replication by using tissues of different chirality. Formation of helical arrangement of molecules in liquid water. The helical arrangement of tetrahedral molecules that occurs instantaneously in liquid water. In liquid water, water molecules are connected by hydrogen bonds and are in thermal motion. In liquid water, the instantaneous atomic arrangement can result in a helical structure. The tetrahedron of liquid water molecules has an ionically bonded region and a covalent region, which splits the energy levels and distorts them to the lowest energy state. In the liquid water, the atomic arrangement of the helical structure shown in Fig. 1 is switched by thermal oscillation. Similarly, silicate silicon oxide is a substance made of a slightly distorted tetrahedron as a component. The silicate has a number of lattice structures, but among them, the alpha quartz of helical structure is the smallest in size and the lowest energy state. Helical lattice structure that works together to generate organized thermal vibration. In a tissue in which tetrahedral molecules are connected to form a helical structure, the tissue of atoms is changed by alternating rotation with the electrical axis as the axis of rotation as shown in Fig. 2. In an environment where there is such a movement, the left-handed L-type and right-handed D-type forms of molecules with the same helical structure may exist at the same time. The tissue of molecules with different chirality can become templates with mirror symmetry. Evolution of molecular tissues of intermolecular bonds. Formation of living organisms by molecules of intermolecular bonds. In liquid water, a wide variety of atomic configurations change instantaneously in the -12 power of 10 seconds. The thermal motion of liquid water has a rotational thermal oscillation of a helical arrangement of tetrahedral units, and there is a region of ascending and descending atoms in the motion. Due to this chain thermal motion, atoms are frequently replaced, and a huge variety of configurations are occurred through trial and error. Properties of organisms that form ecosystems and co-evolve. The molecules of the first proteins were formed in a random process. However, the probability of the formation of a tissue of molecules useful for vital activity is small. Tissues of long-lived molecules increase in population over a long period of time, even if the probability of appearance is small. In the tissue of intermolecular bonds, the molecules present affect the neighboring molecules, and the organization of the original molecule also changes under the influence of the surrounding changes. When there are multiple long-lived tissues, they work together to form a complex molecular structure that increases stability. Since the birth of life in this way, life has continued to co-evolve by forming ecosystems. Where had hydrocarbons of the membrane produced? How had produced the main components of the cell membrane? For the birth of living organisms, a compartment that separates the inside and outside of the cell is necessary. The main components of the cell membranes of all living organisms on Earth are long-chain hydrocarbon molecules C16H34 and C18H38. Its origin is that it was formed on the Earth in the early ancient period when high-energy hydrogen ions from the Sun collided with the atmosphere of CO or CO2 at a high speed of about 500 km per second. The produced small molecules, such as methane CH4, stayed in the sky, and the synthesis reaction continued, and the repulsion between hydrogen ions produced long-chain hydrocarbons. Among these molecules C16H34 and C18H38 were in a liquid state in the temperature range of the Earth's surface at that time and floated on the surface of the water and gathered hydrophobically to form a film as shown in Fig. 3. The current cell membrane has evolved into a bilayer membrane with phosphoric acid and fatty acids on the outside, and a bilayer membrane with hydrophobic lipids on the inside. Permeability of chirality molecule in the phospholipid double-layer membranes. Long-chain molecules that have invaded the bilayer membrane of the cell can pass in one direction because the input direction is reversed in the center and enters the opposite membrane, as shown at the top of Fig. 4. If an oxygen atom is attached to the end of a long-chain molecule, the invading molecule will remain in the membrane. However, as shown in the lower part of Fig. 4, when the two enter the front and back sides of the bilayer membrane, they do not mix, but push each other in the center, forming a double helix like DNA. How did the molecules of the first protein replicate? D-type protein molecule serves as a mold of L-type protein molecules. As shown in Fig. 5, there are two types of amino acid sequences, i.e. left-handed L-type and right-handed D-type. In the environment of liquid water, where the helical structure is frequently formed instantaneously, L-type molds of amino acid units are formed by the surrounding molecules that come in contact with them. The D-type molecule contributes to the replication of the L-type template molecule by constituting the structure of the molecule carrying the L-type amino acid. How had evolved protein replication? If amino acids are matched by replication of protein molecules, replication occurs randomly, and accurate replication is not possible. Therefore, the molecular sequences of complementary bases with different chirality are identified and replicated by an amino acid step. In DNA, which regulates protein replication, the amino acid sequence information is represented by the label of complementary base pairs that have replication functions. Most of the amino acids in the proteins of living organisms on Earth today are L-type, and the sugars in biological tissues are D-type. Evolution on the replication of protein. DNA manipulates the sequence of amino acids in proteins by an amino acid step. In the double helical structure of DNA, the molecular sequence that becomes the template and the molecular sequence that is replicated are matched by staggering them in amino acid steps. In DNA, molecular sequences of the same molecule with different chirality have the same range of separation, and one molecular sequence is a template for the other. Therefore, the identification of amino acids in DNA is carried out by each representative for amino acids in bases. Polymerase produces a single strand from DNA, L-type template mRNA and D-type tRNA. The L-type mRNA has 3 pairs of bases as codons as a label representing amino acids, and the other D-type tRNA has 3 pairs of bases with anticodons, which are amino acid labels provided that the label is a complementary pair. The amino acid molecule carried by the tRNA is added to the molecule of the protein being generated. The replication of DNA indicated by double helix of DNA. The pair of strands in DNA intersects each other in amino acid steps. In Watson and Crick's model of DNA, a double helical structure consists of 2 strands, a leading strand and a lagging strand, as shown in Fig. 6. In the double helix of DNA, in which the direction of proceeding of the helical structure crosses up and down. Since the segmentation of amino acid is the same even for the different chirality, so it can be collated by the amino acid step. The synthetic reactions are performed without mixing the molecular groups of the lagging template chain and the leading template chain, which have different chirality. Reactions to form a double helix of DNA. Constituents of DNA. Constituents of DNA is nucleotide, deoxyribose, nitrogenous base and phosphoric acid. Fig. 4 shows that it is made of. There are 2 types of pair of bases, i.e., adenine, thymine, and guanine, cytosine. How atomic arrangement of complementary base pair becomes mirror image symmetry. The substrate for DNA synthesis adds the following nucleotides to the 3' end of the DNA strand through the medium of triphosphate. As shown in Fig. 7, the direction of proceeding of the replication of the leading strand is opposite for the lagging strand. So, the atomic arrangement of the bases around the lagging strand is mirror image symmetry of the base at leading strand. How complementary bases replicate. Complementary base pairs paired with hydrogen bonds that can be replicated. The groups of atoms of the bases connected to the leading strands of DNA recurve and form a mirror image relationship, which becomes a template for the atomic arrangement of the bases before connection. On the other hand, bases connected to antiparallel lagging chains are connected, but the atomic arrangement of the bases is reversed and does not change. On the left side of Fig. 8, thymine connected to the leading chain is connected to form a mirror image symmetric thymine template but is paired with adenine by hydrogen bonding. Similarly, adenine connected to the leading chain forms a template MER of mirror image symmetrical adenine but is paired with thymine by hydrogen bonding. Therefore, if the opposite strand of base are shifted, adenine or thymine can be replicated in a mirror image relationship as shown in from Fig. 8.3 to 4 or from Fig. 8.1 to 2. As shown in the figure on the right side of Fig. 8, there is a similar relationship between guanine and cytosine. Therefore, if the leading and lagging chains are shifted slightly, complementary base pairs connected by hydrogen bonds can be replicated. How to produce the lagging strand. In DNA replication, the double helix is unwound by DNA helicase, and the lagging and leading strands are replenished with base, as shown at the bottom of Fig. 9. A new lagging strand is added in the downward direction, which is the opposite of the upward direction in which the double helix is unwound. In Fig. 10, they are shown by 6, 5, 4, and 3. There, the starting point of each small region where the direction is reversed is separated by a primer, which are double-stranded DNA. The primer is added to the 3' O to produce a template junction between the Okazaki fragment and the primer. The primer ends at 3, but the primer is followed by a single-stranded lagging template strand, as shown in Fig. 9, so it continues to add helical structures according to complementary base pairs and elongates in the opposite direction until it reaches the double-stranded region. Activities of a life through the replication of proteins. Since the R group of amino acids is hydrophobic, it attaches to the cell membrane and becomes a structural protein. Amino acids are attached along the chain reaction generated in the cell membrane to produce protein molecules, which become enzyme proteins that memorize the chain reaction. Therefore, DNA works to produce proteins that connect the relationship between supply and demand through the activity of states in the supply and demand relationship. Summary. Conventional descriptions of DNA and protein replication have not stated the importance of chirality. If the atomic arrangement of the helical molecule differs by the chirality, the two do not mix, and one becomes the template of the other. In a double helix of DNA, the atomic arrangement of the constituent helical molecules is mirror image of the other, so the two do not mix, and one becomes the template of the other. Therefore, the mechanism of replication of amino acid units that make up DNA and proteins can be clearly explained by the chirality of the helical structure. Thank you for your attention.